All right, hi there AP Bio. I have got some details on an upcoming modeling activity that we're going to have for cell communication. And I'm filming this video because some of you will be out of town while we are doing this activity, but you can still complete the activity with the help of this video. So I'm going to go through the model so that even though you don't have the hands-on portion, you'll still be able to complete your objective. So we are looking at our cell communication activity. There are 5 parts. Part 1 is figuring out the model. So let's go ahead and show you some of those details here right now. So you're going to have, well, you won't have, but this is our model. It is a fancy laminated piece of paper. And at the top of the paper, we have what we are calling our signal producing cell. At the bottom of the paper, we have our signal receiving cell, and a big difference here is that there are these black receptors on this cell. And in the middle of the model is this plastic box. And inside the box are tiny little plastic cups that are meant to line up as kind of like the receiving sort of acceptor site of these receptors. So they're pretty small and things can fit inside of them, all right? So there's also 2 extra little pieces to this kit, and they're packages of beads. So the first part, in part 1, there are tiny little red beads, pink heart-shaped beads, and a larger green bead, and there's a whole bunch of them. You're gonna use these pieces of the beads and pieces of the kit for basically parts 1 through 4. Another part of the kit it says part 6, but I've actually cut out a part. And so this is going to be used for part 5, even though the directions will still say, find the bag that says for part 6 and put them in. So these are the pieces within part 6. So let me go ahead and show you what these beads are going to do. So when the beads get placed in the plastic box in the center, try not to lose any if you are here in the classroom. You close the box, and question number 2 says, tilt your model so that these beads are in the signal producing cell. So you can see here in the model, they're living and they're being produced in this cell, made by this cell. And then the question 2 says, tilt your model so that the signal molecules will travel from the signal sending cell to the receiving cell. So I'm going to flip it around, and if you're lucky, there we go, I got one. You can see that their red beads have landed in the cups, or in a sense been accepted by the receptors. The green ones are too large to do that, and so are the pink ones. So this is going to be used as a model to represent a number of things. In the intro, part 1, you're just kind of figuring out what all these pieces are representing. In part 2, you're going to look at how glucose and insulin will normally function. Part 3, we're going to look at diabetes and how we have problems with blood sugar, glucose, and insulin. Part 4 is normal cell communication. So we're not looking at insulin and pancreatic cells or target cells, but we're looking at like neuron communicating to another neuron. So there's an image in part 4 about normal cell function. So we're kind of representing neurotransmitters moving from one cell to the next. So the kind of cell that we're talking about is going to change in part 4. In part 5, you are going to add some more beads. So let me go ahead and do that. For question number 32, it says to add all the beads. So I'm not adding these little sponge things. That's for another question. But adding all the beads, and you'll notice that the blue and the red beads are the same size. And so I'm going to shake things up a little here. Close my kit. I'll show you. Oh wow, I got multiple binding. So now I've added blue beads that are the same size. You can see that they're also binding in there with the red ones. You shouldn't be seeing 2 at a time, but I can't change it, and I'm not going to mess with it. But red beads can bind, blue beads can bind, and in question number 32, the blue beads are representing something else. So how does this change our interpretation of how these cells are communicating? And lastly, for question 36, we're going to add the foam sponges into the cups. And of course, I've lost one in my moment of doing this. I'll find it in a minute. But in question 36, the receptors get blocked by something. And you can see some receptors that are still open and available can be receiving the signal and binding to them, but other receptors are blocked. So then there's a question in 36 application about what happens if your receptors are blocked by something, what's going to be happening to this cell communication process. Okay? There's one more component to this lab, or this activity, and it's an extra paper called Changes in Receiving Cell Function. Okay? So this document is added on. It's part of the kit as well. And you're going to use these letters A through H on the different cards to make a prediction about how this cell is going to be responding. So this is applicable to questions 13, 15, 16, and 28. I'll say that again. 13, 15, 16, and 28. So I'll go ahead and zoom in closer to these so that you can see them and make the call when you're going to need to say A or E or F or whatever. So in card A, we have a response of glucose getting changed into glycogen by a certain enzyme. In card B, we've got glucose getting changed by an enzyme into fat. Card C is glycogen getting changed to glucose. D is a glucose transport protein closed, so glucose is not able to move across the membrane in D. In E, we have fat getting turned into glucose. In F, we have an open ion channel, so sodium ions are able to move and pass through the membrane. In G, we have a glucose transport protein that is open, so glucose has a doorway to get across the membrane. And then in H, we have the ion channel closed, so the sodium ions are not able to pass through the membrane because their ion is closed, or their ion channel is closed. So those are the components of our cell communication activity, and you can use this video to help yourself answer the questions if you're not in the classroom interacting with the model. Most of it is still answerable with the information from this video. So I hope you have a great midwinter break coming up soon. This should be done before you come back, unless you are out of town in the meantime. All right, thanks, bye.